say that recording in progress thing.
Amen.
Great. Hi everyone. I know some people are still filing in, but welcome to this week's seminar. I'm actually not doing the introduction today. Dr. Reddy is going to introduce our speaker. So honestly, with that, I'll just have you kind of take over. And if there's any questions or anything, feel free. People can put questions in chat. We'll have a little bit of session for Q&A at the end. Otherwise, I'll turn it over to you guys.
Great. Thanks, Olivia. I am really excited to be able to introduce one of my favorite people, Dr. Kevin Feldheim, who is the director of the Pritzker Lab of Molecular Systematics. Is that right? Is that the?
Yeah, that's close enough.
We call it the DNA Lab, the Pritzker Lab, all sorts of things. The Pritzker Lab of Molecular Systematics at the Field Museum of Natural History. So if you've ever visited the Field Museum during working hours, you will see Kevin right behind the windows that face into the DNA lab. And what you won't know is on the other side of those glass walls is like blasting electronica music or something that is very typical of Kevin. Kevin manages this multi-user facility that at any given time can have anywhere from like 10 to 60 people all hanging out doing lots of really cool DNA research. And he does a really good job of facilitating research for others, as well as helping design new tools for research. He is kind of the king of microsats and other ways of examining population genetics, and has been instrumental in developing this for all sorts of species, birds, mammals, fishes, and has over 100 publications as a result of all the cool things that he does. But he also has his own research, which has been a long-term study over 30 years of shark genomics and population genetics in lemon sharks. Are you going to talk about lemon sharks as well?
Actually, I'm not talking about lemon sharks today.
Okay, but I will give a plug out to Kevin's lemon shark research, which is just remarkable because a 30-year population genetic study of any species is amazing, but to do it on sharks, which we know so little about, is really, really remarkable. And from there, Kevin's moved on to looking at all these other aspects of sharks, and some of the really kind of novel and surprising results are what he's going to talk about today in terms of parthenogenesis in shark species. So I'll turn it over to Kevin, and he'll wow you with all this cool shark stuff.
Well, thanks very much, Sush, for that very lovely introduction. And thanks everyone for tuning in today. I'm going to talk today about some of the work that we've done recently, looking at parthenogenesis and how it relates to conservation in elasmobranchs. So before we get started, just a few definitions. The elasmobranchs are actually the sharks, skates, and rays. There are over 1,000 described species, although there are probably dozens of undescribed cryptic species, which actually we've been finding using DNA. So parthenogenesis is a form of asexual reproduction whereby an offspring develops from an unfertilized egg. You can also think of it as a virgin birth, meaning that no male sperm is used. And then parthenote is simply an offspring that is produced from parthenogenesis. Okay. So in typical sexual reproduction in animals, there needs to be a male and a female. And despite this, there are actually several disadvantages to sex. One of them is genome reproduction. So during sexual reproduction, sperm fertilizes an egg. And during the production of sperm and eggs, during meiosis, only half of each parent's genome is carried on to the next generation. And if one of the goals of evolution is to pass on as many genes as possible, this is not very efficient. So just a quick review from your intro bio class. Again, eggs and sperm are produced by meiosis. And in meiosis, only half of the DNA goes into each daughter cell, which is of course how ploidy is maintained during fertilization. When eggs are produced, one cell becomes the egg, but three smaller cells called polar bodies seen at the bottom of the slide here are also produced. Now, usually in a normal situation, these polar bodies are simply reabsorbed by the female. But importantly, as we will see, these polar bodies also contain half of the DNA from the female. Okay. Other disadvantages to sex include finding mates, particularly in species where there's sexual dimorphism. Males tend to be elaborate, while females are drab, blending into their surroundings much better than ornamental males. This is a superb bird of paradise. There's actually a great video on YouTube showing this guy trying to attract this female. And of course, males become so adorned that other aspects like escapability from predators become compromised. Disease is another potential disadvantage to sex. Chlamydia is a bacterium that causes the disease by the same name. And of course, it's an STD that's found in humans, but it has also become a conservation crisis for koalas. Apparently, chlamydia in koalas is a major problem throughout their range. And in koalas, chlamydia can cause infertility and blindness, and red eyes, as seen in this guy here, is a sign of the disease. The situation is so dire that in some parts of Australia, a recent study suggested that animals be euthanized to halt the spread of the disease. Physical harm is another potential disadvantage. In some species, mating is a very violent affair. In mallards, it can be so vicious that the female sometimes dies during the mating process. And in sharks, it's also a fairly vicious process as well, with the males biting the females along her fins and her trunk in order to roll her over onto her back. And if he can do this successfully, then he could perhaps insert one of his claspers into her cloaca and obtain a successful mating. And during mating season, the females can get really torn up, as evidenced by this female here. Females of many species actually have thicker skin than males, presumably to protect them during mating season. This is actually from one of the classic papers in the field of elasmobranch reproduction. This is a cross-section of blue shark with a female on the right and a male on the left. And you could see how much thicker the female's skin is. So one obvious question with all these disadvantages is why even bother with sex? So one of the major benefits of sex is that it creates genetically unique individuals. And as environments tend to be variable, if you create many offspring with many different combinations of genes, it is more likely that some of the offspring will have the right combination of genes to survive in an ever-changing environment. Now, an offshoot to this idea is the Red Queen hypothesis proposed by this guy, Lee Van Valen, at the University of Chicago. And here, organisms have to evolve to keep up with disease, predators, and parasites that are also constantly evolving to become more efficient. And they do this by creating genetically diverse offspring. And of course, the reference comes from Lewis Carroll's Through the Looking Glass, where the Red Queen tells Alice, "Now here you see it takes all the running you can do to keep in the same place." And in fact, that's what we see in nature. Sexual reproduction is the dominant mode of reproducing. But like every rule, there are plenty of exceptions. In asexual reproduction, unlike sexual reproduction, an organism passes on all of its DNA. And it's found in many different types of organisms. Amoeba undergo binary fission, where a single-celled organism divides evenly into two identical cells. Some organisms divide in a different way, with single parents forming an outgrowth or bud. Many yeasts, hydroids, and sponges reproduce by budding, typically in combination with sexual reproduction. Fragmentation is seen in flatworms, algae, and some plants can reproduce asexually by fragmentation. So when an asexual organism reproduces, it makes genetically identical offspring. However, if the environment changes where those genotypes aren't compatible, those individuals can go locally extinct. Okay, so this is a good time to come back to parthenogenesis, a form of asexual reproduction whereby an offspring develops from an unfertilized egg. Now, this mode of reproduction is relatively common in invertebrates, including aphids, roundworms, Daphnia, and of course, many social insects have a haploid-diploid sex determination system where haploid males are produced from unfertilized eggs. In vertebrates, this type of reproduction is rare, but there are several species of whiptail lizards where there are all-female populations which only reproduce by parthenogenesis. Recently, there have been some unnatural cases of vertebrate parthenogenesis, all from captive animals where females are held in isolation. And examples include Komodo dragons, zebra finches, checkered garter snakes, and leopard frogs. Now, in elasmobranchs, the first case of parthenogenesis occurred at this site, the Henry Dorley Zoo in Omaha, where a bonnet head gave birth to one pup. Now, importantly, this female never shared a tank with a male. And so the aquarists, kind of figured that this was a case of parthenogenesis. However, one confounding thing in elasmobranchs is that females can store sperm in what's called the oviducal gland. And so a buddy of mine, pictured here, Damian Chapman, he actually was working on a population genetic study of this species, and he had markers, genetic markers developed. And so he actually genotyped the mother and the offspring, and actually found that compared to the mother, the offspring, the parthenote, was homozygous at every single locus that he examined. Damian and his colleagues also described the second case of parthenogenesis in elasmobranchs. This was from a black tip shark. Now, importantly, both of these cases, only one parthenote was produced, and neither one survived very long. The black tip was actually described during the necropsy of the mother, so it was actually never born. The bonnethead pup was actually bitten within the first week of being born by a stingray, and sadly died shortly after being bitten. Now, that's kind of where our lab came into the parthenogenesis picture. In 2002, a female white-spotted bamboo shark laid 4 eggs. And this happened at the Belle Isle Aquarium in Detroit. Now, Damian didn't have time to develop markers for this species, so I actually developed some genetic markers for this species. And we set out to basically genotype the mother and the offspring. Now, unfortunately, by the time I came into the picture, it was only one mother and 2 of the offspring, because one of the parthenotes stopped eating after about 8 months and basically died from starvation. The other one, I don't know why I always laugh when I tell this. The other one actually jumped out of the tank, and the aquarist found it the next day, and obviously didn't make it either. So the mother and 2 of the pups were sampled, and I genotyped the mother and the 2 pups. And again, importantly, we found that the pups were homozygous across the board compared to the mother. Now, unfortunately, the Belle Isle Aquarium closed its doors, and the 2 pups were actually donated to the Denver Aquarium, and they essentially were lost to science. So we really don't know the outcome of those 2 pups. And our lab has described parthenogenesis in other species, including the brown-banded bamboo shark, a swell shark, an epaulet shark, and a honeycomb ray. Now, it's important to note that all of these parthenotes were homozygous compared to their mother, and also were all female, which lends support to some early evidence that elasmobranchs have an XX-XY sex determination system, much like mammals do. Okay, and so what happens during parthenogenesis, in elasmobranchs anyway, is that one of the polar bodies is used to restore ploidy. And we're fairly certain it's the polar body that's directly next to the egg here, because these individuals are homozygous across the board. If it was one of these other polar bodies to the right, then the individuals would be almost a complete genetic copy of the mother. But since it's this one, this genetic material is almost the same as the genetic material in the egg, leading to almost a complete homozygous individual. There are some exceptions to that. We have found that in some individuals, there are some heterozygous loci, and that's due to crossing over in meiosis 1. Okay. Now, you may recall from your Genetics 101 class that high levels of homozygosity can lead to the expression of lethal recessives, meaning those traits that are normally masked in the heterozygous condition, but can be lethal or at least reduce survivability in the homozygous condition. Now, I don't know if these traits are caused by lethal recessives, but I think we could all agree that they do not bode well for survival. Okay, and of course, the way we determine whether or not individuals are homozygous is by looking at the DNA. Now, I just want to very briefly introduce the genetic marker that we use. Now, this is one way to look at a DNA sequence with the 4 components of DNA represented by different colors. And so at the top of this DNA sequence, you might notice that the G's, A's, T's, and C's are in kind of a random order until we get to an area where AC is repeated over and over and over again. Now, this repetitive element in the DNA is known as a microsatellite. Now, when we use microsatellites to genotype our individuals, we actually don't sequence each locus. What we do instead is we initially find these markers in whatever species we're interested in, and then we develop we develop PCR primers that flank the microsatellite, and we essentially use PCR to amplify the entire region. So in this particular case, this entire region is 125 base pairs, okay? And microsatellite alleles then are represented as number of base pairs, with the main assumption that any difference in size is caused by the number of times this AC repeats. So elasmobranchs are diploid like us, and so they get 1 strand of DNA from mom, 1 strand of DNA from dad. So let's just assume in this case that this allele is from the mother. Let's say the allele from the father has the AC repeated 12 times. The difference here is 10 base pairs. So this other strand would be 115 base pairs long. And we would write this genotype 115, 125. So again, it's important to note that we don't sequence these. Instead, we're using PCR to simply amplify the region of DNA where these microsatellites are. Okay, so we use PCR to essentially amplify every single microsatellite loci at every individual. And microsatellites tend to be extremely variable. And so most individuals that we genotype are heterozygous. So for example, the AC might be repeated 5 and 11 times in one individual, 6 and 10 times in another. We would call these individuals heterozygous. Sometimes the paternal and maternal alleles have the same length. So maybe this individual has it repeated 8 times on both strands. We would call that individual homozygous. All right. So with all that in mind, I want to discuss 2 projects where we found parthenogenesis. The first is looking at parthenogenesis in a wild-caught vertebrate, the small-tooth sawfish. Now, this work was mainly collected by Andrew Fields, who was working on a larger population genetic project of this species. He did this for his PhD at Stony Brook in New York. And then, of course, we have folks that collect the samples for us. Greg Poulakis has been working with this species for a couple decades, and one of his colleagues, Rachel Scherer, have been collecting sawfish from Florida since 2004. Now, the small-tooth sawfish is one of 5 sawfish species. It's the only one found in U.S. waters. All 5 of these species are considered endangered or critically endangered on the IUCN Red List, and all are listed on CITES Appendix 1. And of course, they get this name from this crazy-looking rostrum that have these rostral teeth. And you can imagine what they're used for. Hopefully that's not lagging, but they basically will swim up to some fish and they'll wave that rostrum around. The rostrum also has electroreceptors on it, so they use it not only for obtaining prey, but also for detecting prey as well. And these videos were done by a colleague of mine, Barbara Warringer in Australia. So thanks to her for sharing these cool videos. Okay. Now, sadly for the sawfish, these rostra actually get caught in fishing gear. And so here's a picture of a gill net and a rostrum being caught in a gill net. And of course, as a result, small-tooth sawfish populations have been severely depleted since the mid-1900s. In fact, one study looked at sawfish catch rates from the 1950s to today and estimate only 1 to 5% of sawfish remain compared to the mid 1900s. And so historically in US waters, they occurred in the Gulf from Texas, across to Louisiana and Alabama, across the panhandle of Florida, and then actually up the coast to Georgia and the Carolinas in the Atlantic. Today, however, only two strongholds remain. One in Fort Myers on the west coast of Florida, and another in the Everglades National Park and extending into the Florida Keys. Now, we have collaborators that have sampled both sites, but I'm just going to focus on the Fort Myers region for today. So there are two main sites that Greg and Rachel and their colleagues have sampled in Fort Myers. B here is the Peace River, and a site just to the south is the Caloosahatchee River. And of course, you might be able to tell from this picture, there are a lot of people that live in these areas. Despite that, these juvenile and baby sawfish are actually seen over the mudflats in relatively large numbers. And it's not uncommon to see several of them kind of hanging out together. And not surprisingly, when we genotype these guys, they're actually sibling and half siblings. Okay, so the folks in the field, they catch these sawfish using several methods: gill netting, long line fishing, rod and reel. They tag every animal, they measure every animal. The vast majority of animals in our data set are newborns, juveniles, but we do have some adults, and the adults can get very large, as evidenced by this picture. And of course, they tag all of those animals and sample them for the genetic work. So when I first started this project, we again developed species-specific microsatellites. Again, these microsatellites tend to have lots of alleles with high levels of observed heterozygosity, making them very ideal for looking at things like parentage, sibling relationships, and so on. And so we genotype all of our animals that we capture. And one of the things we do is we run them through a program called Colony. And what Colony does is it basically looks for shared alleles between individuals, and then we'll put them into full sibling groups. It then looks for shared alleles between full sib groups to indicate which groups are actually half sibs. And so from these data, we've actually been able to glean a lot of information from the mating, about the mating system of this species. First, some females mate with several males. This is known as polyandry, one female, several males. Now, we found that 28% of all litters are the result of polyandry by females. Now, this is a fairly low number for elasmobranchs, actually. As Dr. Reddy mentioned, I work on lemon sharks. We found that lemon shark females will mate, I think it's 85% of all lemon shark litters are the result of polyandry. So in lemon sharks, it's actually the rule. In sawfish, it's actually the exception. Another thing we've been able to do is look at individual females and how they are behaving at these two sites. Now, let me briefly explain this figure. Each line represents a female. A diamond above a line indicates that a female gave birth in a particular year. So for example, if we look at line 3, female 3 here, she gave birth in 2004, and then again in 2009. And so we've been able to look at individual females and how they mate at these sites. 34 of these females gave birth at the Caloosahatchee River. 13 females gave birth at the Peace River and only used these two sites. Only 8 females actually used both sites, giving birth in one site in one year and then coming back to the other site and giving birth. But typically, they remain fairly loyal to one site or the other, despite the fact that these sites are so close to each other. Many of these females, the ones highlighted here in yellow, exhibit a biennial cycle of reproduction, coming back every other year to give birth at these sites. There's no example of a female coming back in a consecutive year. Now, we know from another study, a colleague of mine just published something that stated that at least in an aquarium setting, sawfish females are capable of annual reproduction. At least in the wild, though, we have not seen that yet. Now, it's very possible that these females are giving birth in the Caloosahatchee, say, and then going to the Everglades, but we won't know that until we look at the data from the Everglades as well. There are some exceptions to this 2-year cycle, of course. For example, female 1 gave birth in 2004, 2007, and again in 2010. Now, another cool thing we found is that some females, the ones highlighted here in green, have been giving birth at these nurseries for over a decade. Now, we don't know for any elasmobranch species how long females can remain reproductively viable. And so with long-term data sets such as this one, like the lemon shark one that we have at Bimini, can we start to answer some of those questions where we actually need long-term data like these? Okay, as I mentioned earlier, we know that sawfish populations have declined severely from the mid-1900s to today. And when you have fewer individuals in a population, the chances that related individuals will mate with one another actually increases. And so you have a greater chance of inbreeding. Now, one way to test for inbreeding is to calculate a statistic known as the internal relatedness, or IR. And this basically tells us how related an individual's parents are. Now, in a normal outbred population, the values of the internal relatedness should hover right around 0. An internal relatedness value of 0.25 indicates that your parents were related at the level of half siblings. Internal relatedness of 0.5 indicates that your parents were related at the level of full siblings. Now, internal relatedness essentially looks at the amount of homozygosity in an individual. And an internal relatedness of 1 indicates that an individual is homozygous at every locus that we examine. And you all recall what that means. Okay. So when Andrew calculated the IR values of all the individuals in the population, he essentially found what you would expect from an outbred population. The vast majority of IR values were less than 0.1, actually less than 0.2. However, he did find some individuals where the parents were related at the half sib level, and a couple even maybe higher than that, maybe as much as full sibs. But what Andrew also found were 7 individuals where the IR value was either 1 or very close to 1. And when he told us this, we actually didn't believe him. We told him he was wrong. And we actually developed more genetic markers to make sure that this wasn't some fluke. And when we use those other markers, in fact, this IR value only got stronger. And so we were actually wrong and he was right. And so again, if you recall, all homozygous individuals means parthenogenesis. So these 7 individuals were the result of parthenogenesis. Now, it's interesting to point out 5 of these individuals were actually litter mates, and then 2 individuals were unrelated. So we think one female had 5 pups, and then 2 other females had singletons. So 3 females gave birth by parthenogenesis. Now you might be saying to yourself, well, can't these individuals just be homozygous by chance? And I'm going to try to convince you that that's just really not a possibility using some math, which I'm not very good at, but I think this makes sense. Let's see. So if we look at just these first 2 genetic markers, PPE5 and PPE8, the observed heterozygosity are 0.945 and 0.858. So if we look at PPE5 first, the observed heterozygosity is 0.945, meaning that the chance of an individual being homozygous just at this locus is 5.5%, okay? Looking at the next locus, PPE8, the observed heterozygosity is 0.858, meaning about 14% are homozygous at this locus. Now, taken together, an individual just by chance being homozygous at these 2 loci is really low. Only 0.7% of all individuals will be homozygous at these loci by chance. Now, when we combine that over all loci, the chance of an individual being homozygous is almost 0. And so that's why we invoke parthenogenesis to explain these 7 individuals. Okay, so now if we go back and we look at our figure, This is the female that gave birth to the five individuals by parthenogenesis in 2011. She actually came back in 2015 and had another litter, this time by sexual reproduction. Here are the other females. So another female that gave birth to one, initially gave birth in 2011. She came back in 2013 and 2015 and gave birth by sexual reproduction. The other female that gave birth to one pup gave birth in 2004, but then came back in 06, 08, 2010, and 2014, each time giving birth by by sexual reproduction. Now, Greg is very diligent, and he actually takes pictures of all the individuals that he samples. So he actually went back to look at the parthenotes to see if they had any funky mutations. And for one of them, he actually found this. So I was like, oh my God, that's totally, a lethal recessive going on. This is great. This is the result of parthenogenesis. He's like, hold on, cowboy. In fact, this is something that actually occurs a lot because the pups are actually smashed together inside the female. And so oftentimes the rostral will actually be curved just as the pups are born. So Greg actually says that this is probably a normal situation, but it will be interesting to see if we catch any of those parthenotes later in the study to see if they've actually survived. Okay, so the obvious question is, why is this even occurring in the first place? Well, in captivity, the females that use parthenogenesis had either been isolated from males for a very long time or had their males recently removed from their habitat. Now, it's possible that these sawfish are at such low densities that the females cannot find a mate in some years and revert to parthenogenesis as kind of a last-ditch effort to pass on their genes. So since this study came out, I forgot how many years ago it's been, but we've been looking for other cases of parthenotes in our system, and we have yet to find them. Hopefully that's a good sign for these animals. Hopefully the population density is increasing, and so the females are able to find mates moving forward. Okay, so that was one little story. Another project that I've recently become involved with is the STAR project, and STAR stands for Stegostoma tigrinum Augmentation and Recovery, the goal of which is essentially to reintroduce zebra sharks in parts of Indonesia where they've been wiped out. And this has become a very, very large project. So at last count, there are over 40 organizations that are involved with this, from the Indonesian government to several different aquariums to some NGOs, and of course the Field Museum is involved as well. Now, just to give a very brief introduction of the species, the zebra sharks get their name because when they first hatch, they have a pattern that I guess kind of looks like a zebra. And then that actually changes when they turn into when they become adults. They actually have more of a spotted pattern. In fact, in some parts of the world, they're called leopard sharks. This species is found in tropical and subtropical waters of the Indo-Pacific. They're an egg-laying species. And at the onset of this project, I think a lot of folks thought it would be relatively straightforward because this is a very prolific species. One female can lay up to 80 eggs per year. This just kind of gives you an idea of what the eggs look like. So here are the eggs kind of on the edges of this picture with 4 very cute little zebra shark pups in the middle. Now, my work in this project has been driven by these 2 incredible scientists, Katie Lyons on the left from the Georgia Aquarium, and Jen Whiffles on the right from Ripley's Aquariums. One thing I love about being a scientist is you can not only choose what you work on, but you can choose who you work with. And Jen and Katie have been friends for several years, so the fact that I can now work with them is really kind of amazing. Now, this project has afforded us to examine several aspects of the species in captivity. Jen and Katie are leading the first 2, looking at reproductive timing in captivity and mating behavior. Sorry, I can't say captivity, human care. Captivity is no longer PC. Sorry to any aquarists. I don't mean to offend. And of course, I was brought into this project to play the role of geneticist, looking at parental assignment of these animals. Now, before this project started, we knew that this animal, this species, was capable of parthenogenesis because two cases were actually published, one in 2011 and one in 2017. This first one, Robinson et al., described parthenogenesis in an aquarium in Dubai. And actually, this was a case where the female was never introduced to a male. So she basically never had a male that shared her habitat, and she started giving birth by parthenogenesis. Now, the second one was a little more interesting. This took place at an aquarium in Australia, and the female actually initially did have a male sharing her habitat, but once that male was removed, she actually very quickly switched from sexual reproduction to asexual reproduction, and actually had several offspring by parthenogenesis. Okay. Now, one of the differences between our study and those previous studies is we're specifically looking for cases of parthenogenesis and trying to determine how frequently it is occurring. So many of the participating aquariums have been sending eggs to Jen for the past year. So we have Georgia Aquarium, the Shed, Mandalay Bay Aquarium of the Pacific, and the Columbus Zoo. These are the main aquariums. We have other aquariums that are participating, but these aquariums have been sending eggs to Jen over the past year. And Jen, I think, now has dissected well over 1,000 eggs. I mean, it's really crazy, the sample size and the amount of eggs that these females put out. And so in addition to taking several morphological measures, Jen sends me samples to extract DNA. One thing she does is she actually extracts the fluid that surrounds the yolk, and you can see the developing embryo here. And that actually will very frequently yield enough DNA to genotype the embryo. Other times, she actually dissects the embryo out and sends me either a little snip of the embryo or the entire embryo itself, depending on how large it is. This is actually what the yolk looks like. It's got this interesting bluish green hue to it. Now, excuse me, what I do in the lab then is I genotype these animals, and essentially I'm looking for all homozygous individuals to infer parthenogenesis. And although we haven't yet determined how often it occurs, every single one of these females has given birth by parthenogenesis, and really at a very, very high rate. Now, to try to overcome this hurdle of asexual reproduction, several of us met at that mecca of shark research, Las Vegas, because in Vegas, the Mandalay Bay actually has a fairly large aquarium that actually houses a small population of zebra sharks. And so this gentleman right here at the bottom, Lance Adams, Lance is the vet at Aquarium of the Pacific in Long Beach. And Lance has actually been doing some artificial insemination, not only with zebra sharks, but with other shark species as well. And so a bunch of us met there, and Lance, and this is actually Jen as well, they examined both of the male zebra sharks at the Mandalay Bay, and they actually extracted sperm from both of those males. And then the next day we took that sperm to the Golden Nugget, because the Golden Nugget also has an aquarium that houses one female lemon shark. And so Lance and Jen did an examination of the female, did an ultrasound, and then took the sperm from both males and artificially inseminated the female. And so you might be thinking to yourself, well, that solves the issue of parthenogenesis. You're ready to go. But Lance and his team actually just published a paper that actually found that females can still give birth by parthenogenesis even in the face of artificial insemination. And so this figure is from their paper. These arrows indicate when artificial insemination occurred. And despite being artificially inseminated, if we look on the bottom here, a circle indicates that a parthenote was born. A triangle represents an individual was born by sexual reproduction, meaning that the artificial insemination worked. And so if we see there's 1, 2, 3, 4, 5 parthenotes, and only 2 individuals were heterozygous, meaning that the sperm was used by the female. Our team actually also found that female zebra sharks can give birth asexually even when housed with the male. So this was a case at the Shed Aquarium, and Lise Watson is the aquarist over at the Shed. And they actually found that one of the females, they have 3 females and 2 males, one of the females gave birth by parthenogenesis, even though she had reproductively active males available to her. So she gave birth to 2 pups, and it's not like the males were infertile. They actually sired other pups with the other 2 females. So this is really, this is really an issue. And many of these females are actually housed with males, and they're still giving birth asexually. The ones at Georgia, Shed, and Mandalay Bay are all housed with males and are all still giving birth by parthenogenesis. And early results indicate that parthenotes do not fare well. So if we look at growth, the blue bars are heterozygous individuals, or those individuals produced by sexual reproduction. The orange bars are the parthenotes. So you can see that length kind of starts to level off in the parthenotes, whereas the heterozygous individuals are just kind of trucking along just fine. This is also seen in weight, where heterozygous individuals are growing just fine and putting on weight just fine, whereas the parthenotes do not. And of course, most alarmingly, there's not one case of a parthenote surviving. We know that in other species, particularly the white-spotted bamboo shark, other individuals can survive to maturity, but that has yet to happen in these zebra sharks. And so clearly we don't want to put parthenotes back into the wild, and we're trying to ensure we only reintroduce animals that have been produced by sexual reproduction. And so now we're in a waiting game to see what the outcomes of those Vegas inseminations are. And in the meantime, we're continually monitoring offspring produced in other aquariums that are contributing to this project to see if any of the females that are housed with males are giving birth by sexual reproduction. We recently recruited other aquariums from Europe to see if some of those individuals are giving birth by sexual reproduction. And then in the meantime, Jen, Katie, and Lance are also testing sperm storage methods to see if sperm can be shipped not only within the United States, but internationally as well for artificial insemination to increase the chances of sexually produced young. And with that, like in all science, I have lots of collaborators to thank, funding, and then thanks to these folks for the really cool images. And with that, I will take questions. Thanks.
Thanks, Kevin. That was amazing. Really enjoyed all the videos and all that too. You guys are welcome to put your questions in the chat, or raise your hand and ask them, or just unmute yourself and ask. Can I start with one?
Yeah, of course.
So it seems like there's this avenue of research that just developed as people started realizing that this was happening in captivity, right? That parthenogenesis was happening in captivity. I assume this was happening before and people just didn't really think much of it.
Yeah, that's a really interesting question, because it's not like sharks haven't been held for years in aquariums. So my guess is that the aquarist at the Belle Isle Aquarium, the bamboo shark one, he said that when the female would lay an egg, they would just throw it out because they assumed that nothing was in it. And then they started candling the eggs and saw that there were actually little embryos in the eggs. And so they started to save them. Now, why that happened only 20 years ago, I have no idea. So it's an interesting question. And there, of course, it opens all sorts of avenues of research. It's really interesting.
Well, I'll let you answer some of the other questions, but I'd love to hear what your thoughts of how else we can think about using captive animals to study wild populations, right? It's really interesting to do that. I have a question.
There's a question in the chat. What do you feel is the main factor for parthenotes being inviable? Yeah, that's a great question. I mean, the easy answer is we don't know, because we don't. But yeah, it might be the fact that these lethal recessives are being expressed. that's one possibility. Part of the issue is we don't have a genome for the zebra shark yet, so we don't know like what's being expressed and if there are certain areas of the genome that are more important to remain heterozygous, for example. Of course, that's something that we're trying to get. We're trying to sequence the genome, but we won't have that for probably a year.
Anya, you
looks
want to ask your question?
Sure. Hi. Nice to meet you, Kevin.
Hi.
So this is something that I've sort of vaguely heard about, but I'm by no means an expert in, that if you have a high degree of inbreeding, I mean, individuals that aren't actually, full siblings or whatever can effectively look like genetically their full siblings. Do you know what the situation with that is for the sawfish? And I'm also just curious in general, like how do you sort of take that into account when considering their ancestry?
Yeah, we don't, so someone asked a similar question a couple weeks ago when I gave this talk. We really don't. So another, one of the things about this in general, so this doesn't really get to your question, but something that's interesting about this method of parthenogenesis. So it's called automictic parthenogenesis, whereby that one polar body is used to make the individuals all homozygous, right? If one of those other polar bodies were used, the individuals would be heterozygous, and we would have no way of knowing that parthenogenesis even exists. So it's really kind of by dumb luck that we were able to even find that in the wild. But yeah, how that we don't really know whether or not SIBs are mating in the wild. I guess that internal relatedness is the best thing we can use. And with the exception of those individuals, there really isn't a lot of evidence for inbreeding. So hopefully that's a good sign, because again, these animals have been decimated. They are on the US endangered species list, so they're actually illegal to even catch. And they do also, another kind of an aside, they seem to be expanding their range as well. So those two areas are the only two remaining nurseries in the States, but some collaborators found baby sawfish just to the north in Tampa, and also around on the Atlantic side. So we think that means that they're expanding their range. We hope that means that they're expanding their range to what they were seen at historically, but time will tell.
Cool, thank you.
This is somewhat related to Anya's question. Can we tell if the parthenotes are reproducing? Would you be able to like figure that out?
Yeah, so not yet. So we have their genetic tags, right? So these things don't reach sexual reproduction until they're like 12. And so we wouldn't know that for, let's see, they were born 2000, and so maybe, if they were, they would start, we should start seeing their pups. So we have their genetic tag, and what we do with all of the genetic tags that we have from, say, animals that we caught in the initial part of this study, we actually use that as in our potential parent pool for colony. And in the lemon sharks, at least, we found that those individuals that we caught early in the study actually came back and gave birth later in the study. And so we found natal homing for our lemon sharks. We don't know yet if that's happening with these sawfish, because the data set we haven't collected data long enough.
Hey, Kevin, related to Sush's question. So, if you think of your sort of basic biology, evolution, ecology textbook, they come up with these examples of organisms that switch between asexual and sexual reproduction, like Daphnia and so on. And kind of, it's always couched in a ecological resource-based sort of argument, like, asexual reproduction allows you to basically fill available you know, ecological space faster than, you know, sexual reproduction. But I mean, that wouldn't really logically be necessarily the case for chondrichthyans, right? I mean, they're slow reproducers. Yeah, the zebra sharks produce a lot of young, but most elasmobranchs don't,
Right.
and they don't mate they don't reproduce every year. So, and yet it seems to be something they do, right? I mean, phylogenetically now you got it in rays, you got it in sawfish, you got it in multiple, you got it in your carpet sharks. I think there's an example in carcharodonid sharks too, right? I mean, it seems to be, if you had to optimize it, the entire clade has the potential to do it. So, what's your thoughts on that? I mean,
Yeah, it's an interesting question. So, when we published the sawfish study, you have to come up with some BS reason, right? You have to make up a story as to why they're doing this.
Right.
And so we said, well, it's this last ditch effort to, pass on their genes when they can't find a mate. Another thing, like there was one of the bamboo shark papers said, well, maybe this is a way to seed a new coral reef, for example. So evolutionarily, maybe that's why these things happened. Obviously, there's selection to do it, right? Otherwise, it wouldn't be a thing. So we think it's like this last ditch effort. I mean, especially in captivity, human care, where there are no males available to them, and they start putting out these eggs that actually have embryos. Now, the issue, of course, is at least in zebra sharks, none of them survive. So at least to adulthood. So that, I mean, obviously, then why even evolve to do it if they're not even surviving? That, I mean, to me, that's kind of one of the next interesting questions is why are they not surviving to then pass on their genes? Because then it makes no sense evolutionarily to do that.
thank you.
Does anyone have any other questions? Okay, well, Kevin, thank you very much for this. This was really great. I think you overwhelmed us and definitely showed some really cool videos. We were
Great.
wowed by that.
Yeah, good. Thanks, everyone.
Thank you.
Thanks, Kevin.
Thank you.
See you soon, I hope.
Yeah, good seeing you guys.